DNA replication is the process of copying a cell's DNA before division. It occurs in the nucleus of eukaryotic cells or the cytoplasm of prokaryotic cells and requires several enzymes, including helicase, primase, DNA polymerase, and ligase, to ensure accurate copying of the genetic material. Let's start with helicase enzyme, which plays an important role by unwinding the DNA double helix. It breaks the hydrogen bonds between the two strands, creating a replication fork with two single strands. These strands serve as templates for building a new DNA strands. And what you see here are the single-stranded binding proteins to stabilize the unwound single DNA strands during replication by preventing them from recombining again. They keep the strands accessible for enzymes to copy the DNA accurately. And now the second enzyme, which is a primase enzyme, that helps start DNA replication. It makes short RNA piece called primers, which give DNA polymerase a starting point to build new DNA strands. Without the primers, DNA replication couldn't begin. And now, moving to DNA polymerase enzyme, which is really important in DNA replication process. It is responsible for synthesizing complementary strands by adding nucleotides in a 5' to 3' direction based on the template strands according to Chargaff's rule, which states that adenine pairs all the time with a double hydrogen bond with thymine, guanine with a triple hydrogen bond with cytosine. Not only that, it ensures high accuracy through proofreading and correcting errors. What about DNA ligase enzyme? Let me explain this. In DNA replication, the lagging strand is copied in small fragments called Okazaki fragments. This happens because the DNA polymerase can only add nucleotides in one direction, 5' to 3', but the two strands run in opposite directions. On the lagging strands, the replication process occurs in small segments as DNA polymerase work in the opposite direction. Each Okazaki fragment is then joined together by DNA ligase enzyme to form a continuous strand like leading strand. And also, we need to talk about the DNA replication process as a semi-conservative. Why it is semi-conservative? Because each new DNA molecule has one original parental strand and one newly synthesized strand, ensuring accurate genetic information transfer. To summarize it, this figure illustrates the process of DNA replication, showing the synthesis of leading strands and lagging strands. The leading strand is synthesized continuously, moving in the direction of the replication fork as indicated by the arrow. The lagging strand, on the other hand, is synthesized in short fragments called Okazaki fragments, moving in the opposite direction, including several enzymes. The first one, helicase, which will unwind the double helix. We have DNA primase that will initiate the process by adding RNA primer. The DNA polymerase, which will build the complementary, and the DNA ligase, which will seal the Okazaki fragments. I need to highlight something that we have another enzyme called topoisomerase, which will prevent the supercoiling of the DNA. That's it. Let's practice building complementaries. Now, assume the template strand sequence is, and they give us a sequence, the template strand from 3' to 5'. It's very easy to use the Chargaff's rule and build it, but I will highlight a point here. Once they mentioned the direction Also, I have to put it in my complementary. So both of them should be the opposite. So I'll start by 5 prime, adenine all the time, thymine, adenine, cytosine, guanine, guanine. I hope you are doing that with me. Thymine, adenine, cytosine, thymine, guanine, and adenine. Don't forget to put the direction at the end. The other one, pause the video and try it by yourself, but don't forget the direction. I think you answered in the right way. Let me answer it with you. So from 5 to 3 prime, so I will start it by 3 prime, cytosine guanine, adenine thymine, thymine, cytosine, adenine, thymine, again cytosine, cytosine, guanine, then adenine. Please, lastly, don't forget to put the direction. Again, they will run opposite each other. And that's all. Thank you.